Enzymes are widely used in genetic engineering, especially for the manipulation of DNA. Today in this video, I'm going to discuss about manipulative uses of enzymes. Kindly stay with me till end of this video. I hope this video will be helpful for you.
Hello everyone, welcome to my channel Dr. TAPATI'S presentation. Myself Dr. Tapati.
The term biotechnology represents a fusion between biology and technology. The natural processes or age-old practices like preparation of wine, beer, bread, curd are regarded as old or traditional biotechnology. Whereas new or modern biotechnology means all the genetic manipulations, cell fusion techniques, and improvements made in the old biotechnological processes. Genetic engineering primarily involves the manipulation of genetic material, that means manipulation of DNA to achieve the desired goal in a predetermined way. Some other terms are also in common use to describe genetic engineering, like gene manipulation, recombinant DNA technology or rDNA technology, gene cloning or molecular cloning, genetic modifications, or new genetics. In enzyme unit 37, already I have mentioned that according to Triven, applications of enzyme has been grouped into 4 broad categories, including therapeutic uses, analytical uses, manipulative uses, and industrial uses. Already I have discussed about therapeutic uses and analytical uses. Let's know about manipulative uses of enzymes. Most of the enzymes involved in genetic engineering or recombinant DNA technology fall under the broad category of enzymes known as DNA manipulative enzymes. They are a group of specialized proteins that play an essential role in the maintenance and expression of genetic information. These enzymes are involved in the degradation, synthesis, and alteration of the nucleic acids. Within the cell, they catalyze chemical reactions like replication, repair, recombination, and transcription that manipulate the structure or sequence of DNA. Depending on the type of reaction that they catalyze, DNA manipulative enzymes can be grouped into 4 broad classes, including nucleases, ligases, polymerases, and modifying enzymes. Nucleases are enzymes that cut, shorten, or degrade nucleic acid molecules. Ligases join nucleic acid molecules together. Polymerases make copies of molecules, whereas modifying enzymes remove or add chemical groups. The basis of recombinant DNA technology is cutting, joining, and modifying DNA molecules. That means manipulation of DNA molecules in the test tube in laboratory by using DNA manipulative enzymes. Already I told you there are 4 classes of DNA manipulative enzymes. First of all, nucleases, they can be categorized into 2 groups, endonucleases and exonucleases. Second group is ligases. There is no such classification of ligase. Examples of polymerases are DNA polymerase 1, cleaning fragment, DNA polymerase, and reverse transcriptase. Examples of modifying enzymes are alkaline phosphatase, polynucleotide kinase, and terminal transferase. In this video, I am going to discuss about these 4 classes of DNA manipulating enzymes one by one. First of all, nucleases. Already I told you they cut, shorten, or degrade nucleic acid molecule. Nucleases break phosphodiester bonds in between 2 nucleotides and therefore they cut or shorten DNA molecules. Now if you see this figure carefully, you can find that this is single-stranded DNA or RNA. In that case, 2 nucleotides attached through the phosphate group and the phosphodiester bond is present here. And in that case, nuclease enzyme will act on this phosphodiester bond. So there will be breakage of phosphodiester bond, and in this case water is involved. So nucleus is acting on DNA or RNA molecule in presence of water. So water molecule means you can write down in this way. Water means H plus ion and OH minus ion, right? So H plus ion will attach with this oxygen group which is present with the sugar moiety, whereas OH minus will attach with the phosphate group. Now you will get this type of product. This is one product and this is another product. Blue color means this hydrogen is actually coming from water molecule and this OH group is coming from the water molecule. Okay, so you are getting these two products. Now that means suppose your DNA, this is one DNA molecule and due to the action of nucleus, suppose nucleus is acting on this position, then you will get these two products. That means this is length of these two DNA will be shorter than this DNA. So shortening of the DNA molecule is happening. So this is also DNA or RNA may be possible. So in this way, what is happening? Cutting of the DNA and shortening of DNA molecule by the action of nucleases. Already I told you there are two different kinds of nucleases, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA fragments, whereas endonucleases break internal phosphodiester bonds within DNA. Now, if you see, this is one DNA molecule. 5' to 3' exonuclease removes nucleotides from the 5' end. So this is the position where 5' to 3' exonuclease will act at this position and at this position, 5' to 3' exonuclease activity. Whereas 3' to 5' exonuclease removes nucleotides from the 3' end. So that enzyme will act on this position. Now, whereas if you consider about the endonuclease, so this is internal bond, this is internal bond. So these are the internal phosphodiester bonds. So endonucleases will act on these positions. Endonucleases will break or can cut the DNA molecule from these internal phosphodiester bonds. Different types of exonucleases are available. I'm going to give you two examples. First one is BAL31, then exonuclease 3. BAL31 is obtained from Alteromonas aspergillana. It degrades nucleotides from both strands of double-stranded DNA from both 5' phosphate and 3' hydroxyl terminal. Now, if you see this figure, you can find out that this enzyme is acting on this position and this position. That means as it is it has the exonuclease activity, therefore it is acting two terminal ends. It is acting on 5' phosphate end as well as acting on 3' hydroxyl terminal. After the degradation, these are the fragments obtained. In the next step, these are the other different fragments obtained due to the action of BAL31. Now, if you see carefully, you can find out that initially there were 8 nucleotides present in this DNA. Due to the action of BAL31, it is converted to 6 nucleotides. And in the second step, you can see 4 nucleotides are present. That means longer the exposure time resulting in shorter the DNA fragment. This enzyme also possesses single-stranded DNA and RNA endonuclease activity and is capable of cleaving at DNA or RNA nicks and gaps. Overall, if you consider about the BAL31, you can say that it has both exonuclease and endonuclease activity and it is acting on double-stranded DNA as well as single-stranded DNA or RNA. Second type of exonuclease example is exonuclease 3. Source is E. coli. It attacks just one strand of double-stranded molecule, leaving single-stranded DNA as the product. It has 3' to 5' exonuclease activity. It is used for linear template DNA generation for the di-deoxy sequencing technique and staggered ends on double-stranded DNA. Now, if you see this figure, you can find out that due to the action of exonuclease 3, two staggered ends are produced because this enzyme is acting from the 3' end. That means this is one cutting position and this is another cutting position. Therefore, you are getting this type of overhanging at the 5' end of this DNA. In the next step, again, you can find out that single nucleotide degradation is happening and cohesive end or staggered end length is increased. Now I'm going to give you two examples of endonucleases. First one is S1 nuclease, second one is DNase 1. S1 nuclease is obtained from Aspergillus oryzae. It clips only single-stranded DNA, including single-stranded nids in mainly double-stranded molecules. Now, if you see this figure, this is single-stranded DNA due to the action of S1 nuclease. These are the final fragments obtained. In the next figure, if you carefully observe it, there is a nick in double-stranded DNA. So it is acting on the single-stranded DNA in this position, single-stranded nicks in double-stranded DNA molecules. So we are getting these fragments. This enzyme is typically used for blunting the ends of double-stranded DNA or for mapping RNA transcripts. It digests supercoiled plasmids primarily in regions containing inverted repeats, which generally occur in origins of replication. In these regions, the DNA adopts a cruciform structure that H1 nuclease recognizes as single-stranded and cleaves it. It can break supercoiled DNA because it contains single-stranded bubbles. It can also be used to distinguish supercoiled from both non-supercoiled covalent circles and neat circle DNA, both of which are resistant to this enzyme. Second example of endonuclease enzyme is DNase 1. Source is cow pancreas. It cuts both single-stranded DNA and double-stranded DNA. It shows non-specific activity, thus it attacks on DNA at any internal phosphodiester bond. Now if you see this figure, this enzyme, I told you it is non-specific, therefore it can act on any position. Due to the action of this enzyme, these are the final fragments obtained. Now if you consider the next figure, you can see that it is acting on double-stranded DNA. So these are the final products. Final products means you can see that prolonged DNase 1 exposure results in a mixture of very short oligonucleotides. These are the short oligonucleotides and few mononucleotides. Restriction endonucleases or restriction enzymes are very important enzymes required for DNA cloning. These are also known as molecular scissors. These enzymes cut double-stranded DNA only at a limited number of specific recognition sites. Those specific recognition sites contain palindromic nucleotide sequences. Now, what is palindromic sequence? those are actually sequences of base pairs that read the same on two strands in 5' to 3' and in 3' to 5' directions. So in that case, as for example, if you see madam, if you read from left to right, it is madam. Now if you read from the opposite direction also, it is madam. So the madam is the palindromic sequence. So in case of restriction endonuclease, they are only recognizing one type of specific palindromic sequence. In my last slide, we have seen that DNA is one also cards double-stranded DNA, but shows non-specific activity. Therefore, it attacks on DNA at any internal phosphodiester bond. Whereas restriction endonucleases bind to the specific recognition sequence of the DNA and cut the two strands at specific points. More than 900 restriction enzymes have been isolated from over 230 strains of bacteria. That means main source of this enzyme is bacteria, which are mainly used in molecular biology lab. Now, I'm going to give you one example of restriction endonuclease, which is EcoR1. This is very popular name. This enzyme recognizes GATTC sequence. So this is the specific recognition site for EcoR1, GATTC. It cuts the DNA between bases G and A. So cutting position is here and here. G in between G and A, when the sequence GAATTC is present in the DNA, and the enzyme cuts both the DNA strand at the same site. Therefore, due to the action of this enzyme, these are the fragments which are obtained from this process, and you can see that sticky ends are formed. That means overhanging positions are there at the 5' ends of these two fragments. Now see why EcoR1, from where this name is coming. Now E means this is the genus, Co means species. This enzyme is obtained from E. coli. So E means Escherichia, which is the genus. Co means coli, which is the species. So it is obtained from E. coli, R strain, and I is the Roman number. Not I, 1 is the Roman number with the order in which the enzyme is isolated from the strain. Let's know about what is sticky end and what is blunt end. When restriction endonucleases cut the strand a little away from the center of palindrome sites, but between the same two bases on the opposite strands, this leaves single-stranded overhanging stretches at the ends. They are known as sticky ends. Already you have seen EcoR1 example. In that case, it is recognizing GAATTC sequence and it cuts in between G and A sequence, and it is producing overhanging stretches, which is known as the sticky ends at the 5' ends. Now, another example is BAM-H1. Recognition site is GGATCC. It cuts in between GG sequence and it is producing 5' overhanging. That means 5' sticky ends. If you consider about the hint 3, it recognizes AAGCTT sequence and it cuts in between AA sequence. It produces 5' sticky ends. Now, let's talk about the blunt end. Many restriction endonucleases make a simple double-stranded straight cut in the middle of the recognition sequence, which is producing blunt end. So in that case, there will be no unpaired DNA nucleotides on either 5' or 3' strand. That means there will be no overhangs. One example of blunt end forming enzyme is ALU1. It recognizes AGCT sequence and it make a simple cut in between AGCT sequence. It is producing two blunt ends. Whereas if you consider about the HEY3, it recognizes GGCC sequence. It cuts in between GGCC sequence and producing two blunt ends. So from this slide, you observe that EcoR1, BAMH1 and HEY3, they are producing sticky ends, whereas ALU1 and HEY3 produce blunt ends. Nucleases are used in replication process, in base excision repair process, nucleotide excision repair, mismatch repair, double strand break repair, and in homologous recombination process. Let's talk about second type of DNA manipulating enzyme, that is DNA ligases. They join nucleic acid molecules together. DNA ligases are the ligases that join or bind two DNA fragments by forming a phosphodiester bond. And in that case, the covalent linkage of the 5' phosphate group of one chain with adjacent wedge group or 3' wedge group of another is coupled with the pyrophosphate hydrolysis of the cofactor ATP or NAD. In this case, you can see this is the phosphodiester bond formation by this DNA ligase enzyme. Now, they play a critical role in repair, recombination and replication process, especially if you consider about the joining of Okazaki fragments during the replication of DNA. DNA ligase can repair the single strand DNA nick. This is the figure. In this figure, this is the DNA single strand DNA nick. It can be repaired by DNA ligase enzyme. Next, they can join two cohesive ends. Suppose this is one cohesive end, this is another cohesive end. They can be joined by this enzyme in order to get the full DNA molecule. Now, blunt end joining is also possible. So this is one blunt end, this is another blunt end. They can be joined by DNA ligase enzyme. Third type of DNA manipulating enzyme is polymerases. They make copies of DNA molecules. DNA polymerases synthesize a new DNA strand which is complementary to an existing DNA or RNA template. Most of them require primer for the initiation of the polymerization reaction. They add deoxyribonucleotides at the 3' OH group of the growing DNA strand. The primary role of DNA polymerases is to accurately and efficiently replicate the genome in order to ensure the maintenance of the genetic information and its faithful transmission through generations. Now, if you see this figure, you can see this is the primer sequence. This is primer sequence. And this is template sequence. This is template sequence of a DNA. Now, DNA polymerase will catalyze or it will participate in the replication process. So, therefore, addition of deoxynucleotides and new DNA strand synthesis. So, this part, if you consider, this is actually newly synthesized strand. Okay, so in this way DNA polymerase will catalyze the DNA synthesis. Mainly four types of DNA polymerases are used routinely in genetic engineering, namely DNA polymerase 1, clean new fragment, tag DNA polymerase and reverse transcriptase. Let's know about them one by one. DNA polymerase 1 is usually prepared for E. coli. It has dual activity, namely polymerase activity and nuclease activity. Due to polymerase activity, it binds to a single-stranded DNA region or nick of a double-stranded DNA and synthesizes new strand. Whereas due to nuclease activity, it degrades the existing strand as it proceeds. Now if you see the figure, in this case, this is the nick, this is a nick, and this nick is filled up by polymerase activity, whereas these two nucleotides are degraded and resynthesized by nuclease activity, degraded by nuclease activity and again resynthesized by polymerase activity. So in the second figure, you can see this position is the newly synthesized DNA, where these two nucleotides are showing the existing nucleotides, those are replaced. Okay, this one and this one. So existing nucleotides are replaced. Okay, the nuclease activity of DNA polymerase 1 is present in first 323 amino acids of the polypeptide. So removal of this segment leaves a modified enzyme that functions only as polymerase. This modified enzyme is known as clean-out fragment. It can only synthesize a complementary DNA strand on a single-stranded template. It is unable to degrade the existing strand as it proceeds. Now, if you see this figure, this is a nick. It can be filled up by clean-out fragment because of its polymerase activity by using this template. However, in this process, these two nucleotides are not degraded. These two nucleotides are not degraded because nuclease activity is absent in clean-out fragment. So only this is the newly synthesized DNA strand. Newly synthesized DNA strand due to due to polymerase activity of clean-out fragment. These enzymes are used for the DNA sequencing. Tag DNA polymerase. This is the DNA polymerase 1 enzyme obtained from Thermus aquaticus. This organism lives in hot springs. Therefore, most of its enzymes are thermostable, including the tag DNA polymerase. It is the most common enzyme used for PCR amplification. Thermostability of this enzyme makes it suitable for PCR as 94 to 98 degrees Celsius is required to denature the DNA. Optimum temperature for this enzyme is 72 degrees Celsius. Nucleotides are incorporated at a rate of 2 to 4 kilobases per minute. However, it lacks 3' to 5' exonuclease activity. Hence, mismatched nucleotides cannot be removed by this enzyme. Only DNA synthesis is happening. Now, if you consider this figure, this is primer because this enzyme needs primer. Okay, so this is primer and this is the template. And due to the action of tag DNA polymerase enzyme, this is the newly synthesized DNA strand. Newly synthesized DNA strand. Let's quickly know about three basic steps involved in PCR. First step is denaturation, which is carried out at 94 to 98 degrees Celsius. This provides single-stranded template for the next step. So double-stranded DNA is converted to single-stranded DNA. Next step is primer annealing, which is carried out at 55 to 65 degree Celsius. So the primers can bind to their complementary sequences on the single stranded template DNA. In this process, two primers are required. Next step is extension, which is carried out at 72 degrees Celsius. Tag polymerase along with deoxynucleotides, DNA synthesis is happening. That means new strands of DNA is synthesized, and this is the result after one cycle where DNA molecules double. So from one DNA molecule, two DNA molecules are synthesized. Now, if the process of replication of DNA is repeated many times, the segment of DNA can be amplified to approximately billion times. That means 1 billion copies are made. Transcription means DNA to RNA conversion, which is carried out by DNA-dependent RNA polymerase enzyme. But reverse transcriptase is catalyzing the reverse reaction, that means conversion of RNA to DNA. Therefore, this enzyme is also known as RNA-directed DNA polymerase or RNA-dependent DNA polymerase. Using single-stranded mRNA template, it synthesizes DNA or complementary DNA or cDNA that are capable of being inserted into different areas of genome. This enzyme is able to synthesize a double helix DNA once the RNA has been reverse transcribed in a first step into a single stranded DNA. They are used by few viruses like HIV, hepatitis B to replicate their genome and by eukaryotic cells to extend the telomeres at the ends of their linear chromosomes. Retroviral reverse transcriptase has their sequential biochemical activities. First of all, RNA-dependent DNA polymerase activity, then ribonuclease H activity, and then DNA-dependent DNA polymerase activity. Let's see reverse transcription process. In this figure, you can find out that first one is single-stranded RNA, which is the template for this enzyme. In that case, RNA-dependent DNA polymerase activity of this enzyme is producing RNA-DNA hybrid. In the next step, RNase A's activity is degrading RNA molecules. Now you can get single-stranded DNA. In the next step, DNA-dependent DNA polymerase is synthesizing the new strand. Therefore, we are getting double-stranded DNA. In this way, RNA is converted to DNA by reverse transcriptase enzyme. Let's know about few other polymerase enzymes. Polymerase 2 is coded by the Pol B gene and made up of 7 subunits. Its main role is to help in repair and also a backup of DNA polymerase 3. It has only 3' to 5' exonuclease activity. Polymerase 3 is the main enzyme for replication in E. coli. This is coded by the Pol C gene. The polymerization and preparation rate is maximum in DNA polymerase 3. It also has only 3' to 5' exonuclease activity. Polymerase 4 is coded by the DNB gene. Its main role is to help DNA repair during SOS response when DNA replication is started at the replication fork. Whereas polymerase 5 is mainly involved in DNA repair. There are several enzymes that modify the DNA molecules by addition or removal of specific chemical groups. Those are known as DNA modifying enzymes. Let's know about few DNA modifying enzymes one by one. First example of DNA modifying enzyme is alkaline phosphatase, which is obtained from E. coli or calf intestinal tissue. In gene cloning process, this enzyme is used to remove phosphate groups from the 5' end of DNA molecules. As you know, at the 5' end, phosphate group is present in DNA molecule, which is replaced by OH group at the 5' end of that DNA molecule by the action of alkaline phosphatase. This is also used as a reporter gene for identification of secretion signals. Second example is polynucleotide kinase, which is obtained from E. coli infected with T4 virus. Now, in this case, it has the reverse effect to alkaline phosphatase. That means it transfers phosphate of ATP to a 5' OH end in DNA or RNA. In this figure, you can see that 5' OH of DNA is converted to 5' phosphate group by the action of polynucleotide kinase, which is just showing the opposite action of alkaline phosphate. It is also used for the labeling of 5' termini of DNA for Maxim Gilbert DNA sequencing method and phosphorylation of DNA lacking 5' phosphate termini. Third example of DNA modifying enzyme is terminal deoxynucleotide transferase, which is obtained from calf thymus tissue. It is a specialized DNA polymerase that does not require a DNA template in order to amplify DNA. You have seen that almost all of the DNA polymerases need template in order to amplify DNA. But the exception is in case of terminal deoxynucleotide transferase. It catalyzes the stepwise addition of single nucleotides or DNTP onto the 3' end of any oligonucleotide sequence. I have given two figures. One is for the single-stranded DNA, another one is for the double-stranded DNA, where you can see that this enzyme is catalyzing addition of DNTP at the 3' position of single-stranded DNA as well as double-stranded DNA. One of the primary uses of terminal transferase is the tailing of vectors and cDNA with complementary bases, thus permitting the cloning of the cDNA fragments. That means this is helping in the process of cDNA cloning. It can also be used for labeling of 3' ends of DNA fragments. Let's know a few other examples of enzymes which are used in molecular biology. First of all, helicase. It unwinds DNA, as for example in conjugal plasmid transfer method. T4 DNA polymerase enzyme is used in the labeling of DNA fragments for use as hybridization probes. RNA polymerase enzyme synthesizes RNA using a DNA template. Primase enzyme is a special RNA polymerase which makes a short primer required for DNA synthesis. Replicase enzyme is RNA-directed RNA polymerase which is used in replication of some RNA virus. Ribonuclease or RNase. This enzyme degrades RNA molecules. RNase H is a special RNase, that means RNA degrading enzyme which cuts RNA DNA hybrids. It is involved in replication of whole U1-like plasmid. Topoisomerase, this enzyme alters the conformation of DNA, as for example by changing the degree of unwinding or supercoiling. Transposase, this enzyme catalyzes the initial steps in transposition process. Thank you very much for watching this video. Kindly like, share and subscribe this channel. In order to get the notification, don't forget to press the bell icon.